Thank you very much for this opportunity. I'm Kyogo Kawaguchi. I am a biophysicist studying the Department of Physics actually in this university. So our lab in the University of Tokyo is right behind us. And so in biophysics, we are interested in the interface of physics and biology. And as Kazuta sensei was introducing, in physics, we are interested in like maybe particle physics or neutrinos or like gravitational waves, or maybe something a little bit more useful like solid state physics. But physicists are usually like comfortable with like pure or clean or controllable aspects of natural science. And there's like a heavy kind of emphasis on mathematical modeling. From the point of view of physics, if you look at biological materials, they're typically like very complex, they're messy, and there's many components that are unique to biology, like the fact that cells are moving around, and they can also like divide and grow, and they can also die. And those factors kind of make it difficult to model. But nevertheless, I mean, life science or biological materials are the building blocks of ourselves, so we want to understand that from the language of physics as well. So as has been already introduced, so many of the excellent scientists in this audience have already contributed to the understandings and disentangling of this kind of biological materials. But just to highlight a little bit of the research that we've been doing, so we've been interested in how the robust many cells dynamics is being kind of pursued in our body. So this is the top movie is the movie of the live skin in a mouse. And this blobs, the blue blobs actually represent the cells, and you can see how the cells are kind of shedding off in live, and they're being compensated very locally by the cell division. So this is kind of like the key robustness that is going on in our everyday life. And a little bit more on the material science side, we've been culturing these cells called the neuroprogenitor cells, and these have specific structures like the elongated shape, and they are also quite rapidly moving around. And because of this kind of factor of this kind of shape and also the fact that they are moving around, they form these structures called the topological defects, which are pretty familiar to physicists and mathematicians. But what is quite interesting here is this kind of anomalous behavior that these topological defects are where the cells get accumulated or where the cells get repelled. And this could potentially lead to like new material design if we can kind of make these kind of artificial cells to come in these kind of big structures. If you zoom in and look inside our cells, the cells are also not just uniform, simple materials, but they are composed of really complex and intricate structures. And what is really fascinating about the cells is that this is also self-organized. So these are kind of, there's no like designer or some kind of like master regulator that's like controlling everything, but everyone's just playing their role in a spontaneous manner, and somehow they make these like nice segregated structures. So this is the cell nucleus after the cell division, and these kind of colored patches correspond to different parts of the machineries. Now, if you look inside, what is composing these organisms is that there's these, for instance, the chromatin, which is the DNAs wrapped around proteins, and these are obviously very important information materials, and we are now gaining understanding of how these physical properties are interconnected with this information material. And also the proteins are information materials because they have their own sequences and the grammars, if you want, but these are also physical entities because they have very specific interactions that could lead to beneficial kind of functions like the antibodies, but also a little bit more soft interactions could lead to something like the liquid phases or these kind of droplets that would form spontaneously. So now we have a little bit of better understanding about how these could be forming from the point of view of physics as well. In relation to HFSP, I've been really lucky to be receiving the early career grant from 2019. And this was a team with Nicholas Revon in Austria and Shantanu Sin in Broad Institute in the USA. And this was really beneficial for me because I was just about to come back from the US. I did my postdoc in the US and came back to Japan to start my own group in RIKEN. And this particular project was bringing in together stem cell biologists, which is this Nicholas, and Shantanu was an image analysis person, and I was the physicist who was trying to put them together. So this was truly like a new research direction for us, and it was a new collaboration. And also, just to point out, the Human Protease is like a really internationally recognized fellowship and the grants, so that's really nice to emphasize from the point of view of Japanese scientists. I just want to end by saying that we actually have submitted a full proposal recently for this new collaboration with a totally new group, and this is on the color of bird feathers. So if there's any reviewer in the audience, I just want to say hi and finish. Thank you.